Okay then, good evening everyone. Um, Today I'm going to be talking on the topic application of plant tissue culture in pharmacognosy. This is a topic in Unit 3 of Pharmacognosy 1, B Pharmacy, fourth semester. I'll be talking to you about the following topics, which include I'll give you the introduction, then I'll talk to you about the basic requirements for plant tissue culture, then I'll tell you about types of cultures, then application of plant tissue culture in pharmacognosy, which is basically the major topic I'll be talking to you about today. So the introduction, first of all, we have to understand what cultures are and what tissue culture is. So tissue culture, basically, when you hear the term tissue culture, it talks about the process of growing cells artificially in the laboratory. So basically, when you hear something is artificial, you should bear in mind that it's not natural. It was not like that from origin. So it's created out of something. So tissue culture involves both plant and animal cells, but in this topic, we'll be talking only about the plant cells because we're more concerned about that in this seminar. So tissue culture produces clones. Clones, in other words, you can say something like Xerox, like replicate. So Xerox. So tissue culture produces clones in which all product cells have the same genotype. So what is a clone? Clone is an organism or cell or group of organisms or cells produced asexually. So if you talk something is asexually produced, it means it does not involve the fusion of gametes or change in the number of chromosomes. So basically, clones are organisms or group of organisms that are produced asexually from one ancestor or stock to which they are genetically identical. So basically, clones are more identical to what they are brought from. Just like, for example, if you go to the Xerox, if the document you go to Xerox and the Xerox copy will be identical, they have similar things. So this is called clones. So basically, this is what clone is all about. So plant tissue culture is a biotechnological strategy for keeping up and developing plant cells, tissues, or organs on simulated medium under controlled ecological conditions. So simulated medium means artificially. So that's what he's talking about. So it was first presented in 1902 by Sir Gottlieb Haberland. So basically, plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture media of known composition. So basically, the conditions should be free from microorganisms. It should be sterile, and the composition of the culture medium is known. That's why it's artificial. It's not natural because you planned it already. It was pre-planned. So I'll go to the next subtopic, which is the basic requirements for plant tissue culture. Now, hormones such as auxins that include something like indo-3 acetic acid, indo-3 butyric acid, naphthalene acetic acid. These are auxins, plant auxins. Then you can see cytokines, gibberellins like gibberellic acid, abscisic acid, polyamines like putrescine, spermidine, things like that. So these are required for plant tissue culture. Then next is environmental condition, which is the basic requirement. So the three basic requirements for in vitro cultures include the nutrient medium, the aseptic condition, which is sterilization, then aeration of the tissue. Now, in this topic, I won't be telling you in details about the requirements because it's not the topic I'm going to be running about. I'm going to be talking about applications, so I'll be going in details about the requirements. So, just for you to have an overview of the whole scenario. So, in this topic, more often we'll be saying things like explant. So, I'll just give you what explant is all about. So, when you see something like explant, it means explants are small pieces of plants, plant parts, or tissues that are aseptically cut and used to initiate a culture in a nutrient medium. So the term explant can be applied to samples obtained from any part of an organism. So these are called explants. I think you should take note of this so you can understand it subsequently when you go through it, the topic. So I'm going to types of plant tissue culture. Now there are so many types, but I'll just run you through these few, which include seed and root culture, endosperm culture, meristem culture, embryo culture, callus culture, organ culture, protoplast culture, and anther culture, and haploid production. So you can see from this chart, you can see the different types of plant tissue culture. As you can see there, there's seed culture, there's meristem culture, there's protoplast culture, there's organ culture, there's bud culture, there's callus culture, there's cell culture, there's embryo culture. So these are basically the types of cultures, and they have so much more. But we'll just go through this one. I'll not be giving the explanation in this topic. I'll just run through it. So these are the applications of plant tissue culture in this chart. Then I'll be giving you details about it. So basically, in plant biotechnology, the helpful items is a plantlet. So now, what are plantlets? So plantlets are small or young clones produced on the leaf margins or the area stem of another plant. I will repeat that. So plantlets, they are young or small clones. Don't forget I say clones like replicates. So they are young or small clones produced on the leaf margins or the area stems of another plant. So while we're talking about plantlets, plantlets are helpful in plant biotechnology. So the plantlets are utilized for accompanying purposes which include clonal propagation. Now, if you talk about clonal propagation, you're talking about the process of asexual reproduction by multiplication of genetically identical copies of individual plants. So clonal propagation has been successfully applied for propagation of apple, potato, tuberose, and several ornamental plants. So another thing is somaclonal variation. So this is a hereditary variation exhibited among plant cells of a culture. So if you talk about somaclonal variety, is additionally utilized for hereditary variation shown in plants generated from a solitary culture. So solitary means single existing culture, like something that's already been existing and it's just it. So like that. So that's what we mean by solitary. So somaclonal is like hereditary variation exhibited among plant cells of a culture. Another of it is transgenic plants. So if you talk about transgene is a gene that is moved into a living being by hereditary engineering. So the organism that contains a transgene is called transgenic organism. Just like that. So it's transgene. So if I'm containing transgene, so I'm a transgenic organism. So just like that. Just take it like that. So the transgenes can be brought into individual plant cells, and the plant cells can be regenerated from these cells. So these plantlets offer rise to the exceptionally important transgenic plants. So induction and selection of mutations. So you talk about mutagens, then mutagens are added to single cell liquid culture for acceptance of mutation. So the cells are washed and exchanged to strong culture for raising mutant plants. Helpful mutants are chosen for further breeding. So now, in that mutation process, if if resilience is shown, resilience means quick recovery from something. So if resilience is shown to stress like pollutants, toxins, flooding, and so forth, then in increase then the surviving cells are taken to strong medium for raising resistant plants. So basically, if you are growing the thing, if you are growing the mutants, if it's showing the resilience, like quick recovery from pollutants like toxins, flood, and things like that, then these things can be kept in high doses, and then their resistance towards this thing can be used to raise plants that are resistant to things like that. So that one is talking about the application in terms of building the resistance of that cell. So resistance to weedicides. So weedicides are added to culture at first in little doses. When these doses are increased in subsequent cultures, the desired level of resistance is obtained and resistant cell regenerates plantlets and plants. So now we'll go to application of plant tissue culture proper, like the continuation. So plant tissue culture is used widely in plant sciences, forestry, and in horticulture, and the applications include: one, the commercial production of plants used as potting, landscape, and florist subjects, which uses meristem and shoot culture to produce large numbers of identical individuals. Another one is plant tissue culture are used to conserve rare or endangered plant species. So um, to rapidly study the molecular basis for physiological, biochemical, and reproductive mechanism in plants, for example, in vitro selection for stress-tolerant plants. I told you, no? So, like I told about the mutants, they used to like if they show resilience, the quick recovery from pollutants, like I told you before, then those things can be used in high doses. If they are kept in high doses of those things and they resist, like they're able to recover from it, then cells like that can be used to develop resistance in such plants. So this one here is talking about also. So plant tissue culture also can be applied to in cross pollination distantly related species, and then tissue culture the resulting embryo which would otherwise normally die, which is called embryo rescue. So for chromosome doubling and induction of polyploids, and for example doubled haploids, tetraploids, and other forms of polyploids, this is usually achieved by application of antimitotic agents such as colchicine or oryzaline. So basically, plant tissue culture also can be applied in large-scale production of artificial seeds through somatic embryogenesis. So plant tissue culture also can be applied in synthetic seeds, like a somatic embryo is encapsulated by artificial endosperm and artificial seed coat. So basically, this is like an overview of the applications of plant tissue culture. You can go through your textbooks and make references so you can understand the topic more, as it's an important topic, and these are just like the overview. That's it for the seminar. So I think you can just go through the slides because the slides is from I made notes from the textbooks and some from the internet. So in case you are lazy to do those things, I've done it for you. So thank you for listening, and thank you for joining the class, and I hope to speak to you soon. Thank you.